I hope I'm audible to everyone. And as Julia said, I'll be making the case that why we need both morphological and molecular tools if we want exact and accurate taxonomic identification and classification of fungi. So I'll start with a, brief overview over different decades wherein we have identified anaerobic fungi. So as we know that they were first visualized in 1910 and until 1970 for almost 64 years they were not known, even though they were observed by many researchers. It was not known that they are anaerobic fungal or even fungal in nature. So it was only in 1975 that they were discovered officially, and later on, several genera and species of anaerobic fungi were discovered over different decades, initially with the help of only the morphological features, including the ultrastructure analysis. And in since early 90s, the molecular tools, including 18S and later on the ITS, was starting to use. And since 2011, we have also been using LSU in addition to ITS for the identification along with the morphological features. So I'll be giving a very brief introduction that how anaerobic fungi were observed prior to their discovery in 1975. So they were visualized as free-living flagellated organisms, later on also termed as the flagellated protozoan or flagellate parasites, and even named Spheromonas communis and Pyromonas communis by Liebetens. And later in 1913, Brown named them Callimestix frontalis because they were very much similar to the Callimestix cyclopsis, which was a flagellated protozoan. So these observations continued until several decades later on. And in 1966, the Callimestix cyclopsis was identified as a chytrid fungi. So it was still the Callimestix frontalis, which were fungal zoospores. So they were still considered as flagellate protozoa. So they were renamed as Neocallimestix. So it was in 1975 that Dr. Colin Orpin first identified these structures and anaerobic fungi. So what he was visualizing these structures even earlier than that, and he was observing a sudden increase in the number of these flagellate structures after the feeding of animal. So at least within 1 to 3 hours. So in this particular work, he realized that these structures are released from the sporangial-like structures, and later on they germinate to form a vegetative thallus, wherein there was a sporangium on one side, and later on this sporangium it ruptures, and then these flagellate structures that many researchers have been visualizing over several decades, they are released. So this convinced Colin Orpin that these structures are not flagellated protozoa as was the case earlier, and then they are definitely a phycomycete fungi. So in the very next year, then another anaerobic fungus was identified as Pheromonas communis, and this particular fungus had uniflagellate zoospore as against the polyflagellate zoospore in case of Neocallimestix frontalis. And as against the filamentous structure of the rhizomycelium, as shown here, this particular fungus has had bulbous type of mycelium. So even and later on in the next year, 1977, another fungus which had uniflagellate zoospores but had the filamentous vegetative type structure similar to Neocallimestix were identified and named as Pyromonas communis. So in 1977, again the chitin was observed in the fungal cell wall, and it proved conclusively that these are actually anaerobic fungi. So until and once the all and importantly all these three genera were monocentric in nature, and it was not until the next decade that the different type of anaerobic fungi, which were polycentric in nature, were identified. So surprisingly, they were all found within 2 year gap. So the first one was named Neocalimastix joanni in 1989, and this was a polyflagellate anaerobic fungi, and it was identified as a species to Neocalimastix because of presence of polyflagellate juice spores. In the same year, another novel genus Orpinomyces bovis was identified. However, later on, both these were found identical, and they were named as Orpinomyces joanni. In 1990, Anaeromyces micronatus, another polycentric anaerobic fungus, was discovered, and within a gap of few weeks, Ruminomyces elegans, which was structurally similar to Anaeromyces, was also identified. And based on the precedents of earlier publication, it was also named as Anaeromyces. So both these anaerobic fungi were structurally very similar. So later on, most of the researchers also agreed that they are actually the same species and not the distinct ones. So after a gap of another decade, the another bulbous anaerobic fungi was identified, and this one, but this one had a polycentric type of growth and had multi-sporangiate thallus. So this was named as Cathalamyces aberrancies. So if we see this, so there were almost 25, 26 years it took to identify first six genera of anaerobic fungi, and most of them were based only on the morphological features. So, if we want to understand the morphology, development, and taxonomic classification of anaerobic fungi, then we first need to understand the life cycle. So the life cycle of anaerobic fungi obviously differs, but it mainly, in all of the cases, it starts with a free juice spore. This juice spore can be a uniflagellate, wherein there may be one to even up to four flagella per juice spore, or it may be a polyflagellate juice spore, where there may be from 8 to 15 or 17 flagella per juice spore. So these juice spores, once they are released into the rumen or the medium, they sense the substrate and then they move to the substrate with the help of chemotaxis, and then they sense the water soluble carbohydrates or phenolic acids, and then they go and attach to the substrate. So once they are attached to the substrate, they shed the flagella as they no longer need it, and this structure is known as juice or cyst, and the process is known as encystment. So once this juice pore cyst is attached to the substrate, it starts germination. So there are mainly two different modes of germination and development in case of anaerobic fungi. So first is monocentric. So monocentric refers to the single reproductive body, mono and centric. So it means from a single juice spore, only a single reproductive body is produced. So this monocentric development is again of two types. One is endogenous and another is exogenous, as shown here. So in case of endogenous, the nucleus of the juice spore cyst remains within it and it doesn't migrate. So once the rhizoids are formed, this juice spore cyst itself enlarges and forms a sporangium. So that is why it is known as monocentric endogenous. While in case of exogenous development, there is a bipolar germination. On one side, there are rhizoids, and on another side, another outgrowth which is known as sporangiophore from juice spore cyst develops, and the sporangium develop at the end of that particular structure. So in that case, that nucleus had to move from the juice spore cyst to the sporangium, and that is why it is called as the monocentric exogenous type of development. Now, since this nucleus remains within the sporangium and it doesn't migrate to the rhizoidal structures, So the rhizoids of monocentric fungi, they are anucleated, and this type of development is known as determinate development. So in another case wherein the polycentric type of development happens, the nucleus of the zoospore cyst, it migrates to the rhizoids, eventually making all the rhizoids nucleated. So this type of development, since the nucleus migrates from zoospore cyst to the rhizoids, so it is always exogenous and all the rhizoids are nucleated. So this type of development is uh, non-determinate, and any of these rhizoids can then later on produce multiple sporangia. So this type of development is known as polycentric exogenous type of development, and since multiple sporangia are produced from a single zoospore, this development is polycentric. So once this sporangium or sporangia have developed, after various nuclear divisions, then zoospores are produced within these sporangia, and then these zoospores are released by way of different mechanisms, and again this entire life cycle starts. So although this particular life cycle I have discussed for filamentous group of fungi, similar development happens even for the bulbous ones. So here we can see, as I mentioned, so the entire taxonomic classification of anaerobic fungi, especially in the initial years, was and still even today is based on these characters, which may be monocentric, endogenous, or exogenous. It may be both, or even one in case of monocentric. And polycentric, as I mentioned, it is always exogenous. The rhizomycelium may be filamentous or bulbous, as we can see in these structures. These are the bulbous anaerobic fungi. So in case of monocentric filamentous, it then depending on the zoospore flagellation, if it is a polyflagellate or uniflagellate, the genus or genera are characterized. So a monocentric filamentous polyflagellate culture is Neocalymastics, while the uniflagellate one is a Pyromyces. In case of bulbous, the zoospore flagellation is always uniflagellate and it's Cecomyces. While in case of polycentric, again, if it's filamentous, it can be both uniflagellate or polyflagellate. And if it's uniflagellate, it will be Anaeromyces. And if it's polyflagellate, it will be Orpinomyces. Similar to the monocentric one, the bulbous here also is a uniflagellate and it's a Cathlamyces. So the correct taxonomic classification of at general level was pretty straightforward. But what happened eventually for species classification, there were many features that were used. The most common was the variations in the shape, size of the sporangia or in the rhizoidal structures. Also, in few cases, how the zoospores are liberated from those sporangia, if it's through apical pore or it is through dissolution of that particular sporangium, and also on based of several ultrastructure differences, and even in few cases based on the physiological differences in fermentation product or their ability to utilize some sugars or presence of some metabolite in the fermentation. So in neocalymastics, at that time, there were four different species that were identified. However, later on, several researchers studied them in detail, and all of them agreed that they are actually the same species, and neocalymastic has only one species. In case of Pyromyces, again, there were many distinguishing features. Some of them were unique, and there was no consensus as to whether they are same or different. For Cecomyces, there were two species, and they were all again agreed to be the same. In orpinomyces, the microscopic or morphological difference is very substantial, and they were agreed to be two different species. While in Anaeromyces, as I mentioned earlier, they were similar looking, and even though they were never compared together, it was kind of agreed that they are the same species and there is only one species in Anaerobic. So these are some of the photographs I have where I am showing different structures. And this is in case of Pyromyces. Here we can see that different rhizoidal constrictions, presence of these subsporangial swellings, and the dissolution of sporangial wall is considered as a distinguishing feature in case of Pyromyces combinates. While in case of another species, it is the constriction at the end of sporangium and at the beginning of the rhizoid, which led to the its characterization as different species. In Pyromyces spiralis, it is the rhizoidal structures that are highly spiralated, and in case of Pyromyces minutus, it was the small size of sporangia, and also the juice for liberation through the apical pore, leaving a cup-like structure. In Pyromyces may, it was these multiple papillated structures, while in case of Pyromyces citroni, it was the ability of this particular fungus to produce multisporangiate thallus. As we can see, there are two distinct sporangium. Later, another Pyromyces species, Polycephalus, was published, and however, the validity and identity of this particular fungus is still debated. So as we can see, there were many variations, and these variations were used for identification, characterization of these different species. So, however, in our work, we what we realized that and this Pyromyces species, they vary a lot. They show a lot of pleomorphism in case of their sporangial shapes, sizes, and they may not be even if they you grow them in a same genus, same medium, or if you grow them in a different medium, the structures are always varying. So here we can see this is the same strain and grown in same medium. So we can see the same strain showed various type of morphologies. Now, if we see, suppose this is a multisporangiate structure, this is also multisporangiate. So if we go by the yeah, even though it is the same strain, if you go by the morphology based identification, this particular strain will be identified as Pyromyces citroni, as in this case, and also this is the papillated structures in these cases. So again, even if so these features are not consistent and cannot be used, but they were used. So even in case of juice for liberation patterns, we can see in this slide. So this is the same strain, and it showed multiple juice for liberation patterns. So here we can say the juice pores have liberated and only this cup-like structure has left. This case, the juice pores have left through the apical pore. Here, the sporangial wall has dissolved even before the juice pores are liberated, and again the apical pores. So the same strain showed multiple ways of juice pore liberation. However, these features were used to classifying different species in case of Pyromyces. And similar sporangial variations were also observed in case of Neocalymastics. So in case of these monocentric genera, these variations were highly pronounced in shape and sizes of sporangium. In case of polycentric ones, what we realized and also documented, the issues are slightly different. So if there are clear sporangial or juice pores in the if the culture is producing them, then we can easily identify Anaeromyces, especially by their ability to produce these constricted or beaded rhizomycelium, and also they produce this beak-shaped sporangium where you can see this pointed apex. However, in sometimes they also maybe at different stages of development. These are not very pronounced. The biggest issue in case of identifying Anaeromyces is their inability to produce either the sporangia or the zoospores after multiple subcultures. The same issue was also observed in case of orpinomyces. So as I mentioned, if the sporangia are produced, we can easily identify and differentiate orpinomyces into its two species based on the presence of the sporangia. If it is intercalary, it is orpinomyces intercalary, or if it is at the terminal end of sporangiophore, it is a orpinomyces joini. However, in this case also, after several subcultures, what we have realized, this particular genus stop producing sporangia. So this is most likely because they have nucleated rhizoids and they don't need presence of sporangia or the zoospore as a mean to continue their life cycle. So maybe they don't need it. In case of Cathlomyces, especially when you grow them on a complex substrates like this is rice straw in these particular images. So even though they produce these bulbous rhizoidal structures, they also show filamentous kind of growth. So like here we can see it's a sporangium at the and this is sporangiophore and in these cases also. So such structures alone, if we use them to identify genus or species, may lead to confusion. So herein I have shown different zoospore flagellation. So if the zoospores they are present, it's very simple to identify them if they are polyflagellate, biflagellate, or uniflagellate. However, the shape and size of these zoospores they it also vary even within the same strain. However, many times there are many structures which resemble like a zoospore, but it is not. For example, in these two cases, we can see this is a zoospore cyst which is germinating. So this may give its identity as if it's a polyflagellate zoospore. While in some other cases, if these all flagella they stick together, even if it's a polyflagellate zoospore, it may give the identity as if it's a uniflagellate zoospore, and similar in these cases also. So, fluorescence microscopy is another technique for morphological identification, which is pretty straightforward, where we can easily see the nucleated rhizoids in polycentric cultures and absence of nuclei in monocentric cultures. However, for example, in this particular case, this is a Pyromyces species. However, we are seeing a structure similar to the nucleated rhizoids. So we assume that these structures are actually the spores resting structures in the life cycle of anaerobic fungi. And even though they have not been fully identified or confirmed yet, but these look like the spore-like structure and they heavily contain these nuclei. And such structures may also give false assumptions for identification purposes. So based on these multiple variations that we have observed in different uh, type of these tools, so there was a need to use molecular methods, especially the bio barcode markers for the identification purposes. And here we can see this is the ribosomal RNA gene operon. And similar to other eukaryotes, it has 18S, then ITS, which is internal transcript spacer 1, 5.8S, ITS2, and LSU. So the taxonomy, the molecular taxonomy, or the marker which was first used was 18S, and since it's long, it was only the partial. And later on, it's complete ITS, which comprise either ITS 1 plus 5.8S or ITS 2, especially in case of pure cultures, or only the ITS 1 or only the ITS 2. For this 28S or large subunit ribosomal DNA, since it's quite long, it is only the 5' region or D1, D2 region of this particular marker that is being used. So the first use of 18S was in 1991, and then they in this particular study, four different genera of anaerobic fungi: Neocalymastics, Pyromyces, which was then known as Pyromonas, then Spheromonas, which is actually Cecomyces, and Neocalymastics joannae means it's Orpinomyces. So Neocalymastics, Pyromyces, Cecomyces, and Orpinomyces, they were sequenced for their 18S rRNA gene. However, the results showed that they are very much similar, even 99.6% similar in the Pyromyces and Orpinomyces, while the Pyromyces and Orpinomyces were 99% similar to the Neocalymastics. So this study and then again subsequent studies by many other researchers, they validated these findings this particular region was found unsuitable for studying the taxonomic relationship within anaerobic fungi. So later on, the ITS, as I said, either ITS complete or ITS1 or ITS2 region was used, and this region is still being used for identifying. But what we have realized over different years of our experience, this ITS is suitable for genus level differentiation, but not very much suitable at the species level. Also, there are many other issues related to use of ITS, especially the size or the length variability. And the biggest one is the presence of intra-individual polymorphism or intragenomic variations. So since all these regions are multi-copy genes, so in case of this ITS, there is lot of this intra-individual polymorphism, and it is as much as 13% as shown in one study by Callahan and workers. So it also makes a problem for direct sequencing of the amplified PCR product, and most of the times we need to clone it to get a clean sequence. And also, at that time, we realized that there are many sequences that are submitted to NCBI, which have been misidentified. So even though morphologically they look different, the sequence, the blast search at NCBI used to give us different results. So that we realized that there is a need for a different biomarker for taxonomic analysis of anaerobic fungi. So for this purpose, we selected the D1, D2 domain of large subunit ribosomal DNA. The same region was also used earlier by Fligrova and co-workers, and in one paper by Hosner in 2000 for RFLP based differentiation. However, at that time there were no sequences for LSU submitted at the NCBI. So we analyzed the sequences and we found this intra-individual polymorphism in the ITS region, as we can see here. While of all the sequences we had analyzed for LSU, there were very little genetic heterogeneity within the sequence, and we used to get very clean sequences. So later on, we analyzed the LSU region, and we found that this region can easily differentiate between these two different species of Orpinomyces, which was not possible by even using the ITS region. And here in this phylogenetic tree also we can see two distinct clusters for both the species. So this again proved that LSU is suitable for even up to the species level differentiation and classification of anaerobic fungi, and the same hold true even for a recently published new species of Anaeromyces, wherein two distinct clusters were produced by LSU region. So this is a just a representation wherein whatever sequences we had, we aligned them over their length, and we can see that they are the sequences. Even though they are there is a lot of similarity, there were enough variations within these sequences in case of different genera and even species. And using these similarities and variations in the LSU, we developed specific primers for the different genera, Anaeromyces, Neocalimystis, Spiromyces, and Orpinomyces, and even for the specific species of Orpinomyces. So these primers could specifically amplify only these targeted genre, and I'm not sure if they still can be used, but at that time, whatever genre were known, they were successful. Those variations we again targeted to find out if there are some restriction enzymes that can in one go differentiate between most or some genre of these anaerobic fungi. And surprisingly, we found that if we do a codigestion of ALU and HINF1, it could differentiate between all non-genera of anaerobic fungi at that time. It was in 2012. So this region really proved useful, and it was very successful and helped in the clear-cut taxonomic identification, classification, and differentiation between different genera and species. So now moving on to that, now why do we need both morphological and molecular tools even today? So I'll just discuss these two cases. First is Otomyces, which was isolated from a camel, and the another one is Bacharomyces. So in Otomyces, if we see the microscopic features, it produces uniflagellate zoospores and it's a monocentric thallus. And even if you see, these are the constrictions at the end of sporangiophore and at the beginning of the rhizomycelium, which is all similar to Pyromyces. However, some other structures like these swellings in the hypha, or which look like intercalary sporangia, which are present in Orpinomyces intercalaris. So the microscopy suggests that they are most likely Pyromyces, but these structures also confuse if it's a Orpinomyces intercalaris. Similar was the case for Bacharomyces, which showed uniflagellate zoospores, monocentric thallus, and again these constrictions at the base of sporangiophore. So the situation proved even difficult when we did the blast search for sequence similarity for both regions, and they showed highest sequence similarity to Anaeromyces. Now, if you see, they are both, even though Anaeromyces is also uniflagellate, they are also uniflagellate, but there is clear differences in their development type, which is monocentric, and Anaeromyces is a polycentric. So this clearly told us that they are distinct genera, nothing related to either the Pyromyces or the Anaeromyces. Later on, this phylogenetic analysis using both ITS and LSU region also showed clear-cut cladding of our strains and helped us publish these two as two different or novel genera of anaerobic fungi. So the same hold true for the recently discovered genera. If we see, it took almost 25 years to discover the first 6 genera, and in this table I have shown all the newly described genera of anaerobic fungi. So within in the last 5 years only, we have discovered 13 new genera. So the same case is here in the identification of these genera as well. For example, Pecoromyces, it also had uniflagellate monocentric filamentous morphological features, but and this showed its similarity with either Pyromyces or at that time Otomyces and Bacharomyces was discovered. However, its sequence similarity was with Orpinomyces, and that is one of the reasons that this particular strain, which is C1A, was published as Orpinomyces species for few years. A similar situation was there for Pharamyces, which also had monocentric filamentous thallus, but the zoospores were polyflagellate. So it means it is similar, structurally similar to the Neocalymastics. However, this particular genus had highest sequence similarity with Pyromyces and a few sequences with Neocalymastics. So similar to with the other recently described genera also like Libetenzomyces, even though it was similar to all monocentric uniflagellate genera, but it was similar highest sequence similarity was with Anaeromyces. And even in the recently described genus, the same is the case. So we can, based on these observations, we can see that if we go by the morphological identification tools alone, we end up identifying them wrongly. And if we go by the molecular or sequencing tools alone, again we are going to end up identifying them wrongly. So it is absolutely necessary to use both morphological as well as the molecular tools to correctly identify these different anaerobic fungal cultures. Thank you.